During the latter half of the 19th century, the notion that all organisms were made up of cells was beginning to take hold. This was due in large part to the development of the microscope. There was one type of tissue, however, that didn't seem to follow this rule: the brain. Cells had been observed here, but the mess of fibers meant there was no clear notion that the brain was made up of discrete cells. In the end, it was the development of the Golgi stain that allowed scientists to see individual neurons are connected to each other. And this idea that the puzzle of the brain can be solved by looking into the brain with more and more detail captured the imagination of neuroscientists for the next century, with the development of more intricate microscopes, MRI machines, and fluorescence imaging, letting us view the brain with more and more complexity. And yet, while all of this development has gone on to improve how we look at the brain, our ability to control the brain has always lagged behind. That was the case, at least, until around 10 years ago, when a group led by Zhao Huapan and another group containing Ed Boyden and Karl Dieseroth kick-started the field known as optogenetics. Optogenetics uses light-sensitive channels taken from algae. These are channels that can open in response to light and allow current to flow through them. The channels are inserted into membranes of neurons. That means that when the neuron is exposed to light, we can control it by either making it fire or stop firing. And this was a massively influential approach. Suddenly, there was a way to selectively control specific groups of neurons in the brain using only light. In contrast, the previous approach was to simply pump electric current into neurons, which is like having a delicate musical instrument and only being able to play it with a sledgehammer. The new optogenetic approach was like being able to pick out individual notes with absolute precision. Like a piece of music, the brain has its own rhythms. There are repetitive patterns of activity. We have peaks, where many neurons will fire at once, and troughs, where activity is lower. Given that all this activity changes from moment to moment within the brain, we can only really be sure that our manipulations are true if we exert our control with reference to what we can see. This is what closed-loop optogenetics aims to do. In closed-loop optogenetics, the light that we use to control the brain is in turn controlled by the activity of the brain. Take an experiment by Siegel and Wilson, who are studying spatial memory in the brain. Spatial memory is dependent on a region called the hippocampus. The hippocampus has its own rhythm of activity called theta rhythm. This means that activity rises and falls every few times every second. By listening to this rhythm, Siegel and Wilson were able to use the light to stimulate the hippocampus at different sections of the rhythm. For instance, they found that when they stimulated at the peak of activity, the peak of the rhythm, they improved spatial memory, but only when they were stimulating when the mouse was learning. When they stimulated the trough of activity, they improved memory again, but only when the mouse was demonstrating its learning. This suggested that different phases of the theta rhythm were involved in different processes of spatial memory, namely encoding and retrieval. And this finding was only possible because they were able to react to the rhythm of the hippocampus in real time. Another application for closed-loop optogenetics is in the prevention of seizures. Seizures are basically when the activity of the brain goes into overdrive. In a paper by Paz and others, 2013, they looked at seizures that were caused by overactivity in a part of the brain called the thalamus. Using light, they optogenetically inhibited the thalamus, and they found that the rats that were previously prone to seizures were prevented from seizuring. However, they wanted to take it a step further. If we inhibit a region of the brain constantly, that may not be very useful for treating a disease, since that part of the brain is probably useful for something. In the case of seizures, you only want to inhibit that part of the brain when a seizure is happening. Inserting electrodes into the thalamus to detect activity, Paz and others were able to listen for and prevent seizures before they happened. One interesting application of being able to react and control to the brain in real time is the ability to use the activity in one part of the brain to control activity in another part of the brain. This approach was used by Atherley in 2018. They asked whether they could reinforce certain patterns of activity by rewarding the brain every time they noticed this pattern of activity. They inserted electrodes into the motor cortex and recorded from a group of neurons. They were looking for a certain pattern of excitation that these neurons as a group would carry out. When they detected this pattern occurring, they optogenetically activated one of the reward centers in the brain, the ventral tegmental area, or VTA. What they saw was that after rewarding these patterns of activity through the stimulation of the ventral tegmental area, these patterns of activity were more likely to occur again. It was as if they trained the brain. All of this was done purely inside the brain without eliciting any behavioral change. This kind of control has the potential to treat many kinds of disease. Take addiction, for example. If we can spot the patterns of activity that are associated with addiction, we could train the brain to stop producing those patterns. In fact, wherever there is a pattern of activity associated with a disease, we could, in theory, prevent that activity and potentially cure that disease. These are three very different ways closed-loop optogenetics can be used for both basic and applied neuroscience. It can be used to break down the temporal separation in the brain's processes. It can be used to detect changes in the brain and react to them to prevent seizures. And it can be used between brain regions, hijacking the brain's reward system in order to train the brain towards certain patterns of activity. While these are three interesting examples of the way closed-loop optogenetics can be used to study neuroscience, they are only one of a small number of closed-loop studies that use brain activity to derive control. One of the potential reasons for this comparatively small area of research is that optogenetics is fairly new, and this is a step more complicated than doing simple optogenetics. Take on top of that the fact that we often use light signals to see the activity of neurons, and the use of optogenetics here can be difficult since there may be crosstalk between the measuring and control methodology. Nonetheless, the field of closed-loop optogenetics is growing, and its full potential will only be revealed as time goes on.